This project is about investigating how mycotoxins production is affected by the environmental factors for ergot and Fusarium head blight. Mycotoxins are toxins produced by pathogenic fungi such as ergot, which produces ergot alkaloids, and Fusarium head blight that produces nivalenol. If mycotoxins are detected in a concentration of 0.5 g per kg, then the crops are not fit for human consumption and therefore either get tossed or used for animal feed. Unless they hit a separate threshold, then they are not fit for livestock consumption either. This is due to mycotoxins causing teratogenic, carcinogenic effects, and in large enough doses can also cause death. Due to the legal levels of allowable mycotoxins within a crop being so low, this project aims to see whether changes in the environment can affect the concentration of mycotoxins that are produced during infection and during sexual production within the soil, which is where mycotoxin production is at its peak for both ergot and head blight. So, to introduce the science behind this, the next couple of slides will go through the disease cycle of both ergot and head blight. Ergot infection begins when ascospores from the wind land onto at-risk wild and forage grasses and crops such as barley during spring. Next, the stigmas of these plants trap ascospores, which leads to infection. These ascospores can infect the ovaries of crops within 24 hours of exposure. During exposure, the ascospores develop hyphae which colonize the ovary and will grow into the ovary axis, henceforth mimicking pollination. Next, the infected ovary grows a stroma that produces honeydew, which attracts pollinators to the crop, which further increases the spread of infection. Other ways this infected honeydew can spread to other plants is through rain, farm equipment, crop head to crop head contact, or splashing to nearby crops. This process continues until the sclerotia matures within 4 to 5 weeks, replacing the seed for a black cereal head fungus. Hence, conditions which delay pollination, such as cool, wet weather, increases the spread of ergot. This infection process is repeated. When infecting cereal crops, a sclerotium is formed, which is a white-like mycelial tissue that contains dark pigmented storage cells for energy, and the sclerotium also protection from UV damage and desiccation, amongst other environmental conditions that could prove harmful to the ergot. During harvest, the sclerotia is harvested together with the grain, whilst some falls into the ground, making the next set of crop grown on the field susceptible to ergot infection. Cereals crops such as barley have an increased likelihood of being infected through a secondary inoculum as opposed to the primary, from nearby infected wildgrass fields. However, resistance to ergot infection begins within a few days after fertilization occurs, therefore meaning that infection occurs just before fertilization occurs. Whilst on the ground for at least 25 days within soil temperatures of 0 to 10 degrees Celsius induces vernalization. This transforms the sclerotium to mushroom-like stalk growths known as the stomata. This is the part where sexual dimorphism starts, with the spores as female Ascogonia and male Antheridia start to develop within the dikaryotic ascogenous hyphae. That causes the meiosis of sexual fruiting bodies, henceforth causing the production of parathecia. These are filled with asci that contain 8 ascospores. This typically occurs during times of high rainfall or high soil moisture, which additionally causes stroma formation as well as ascospore production. During such moist conditions, ascospores are ejected out to the air to act as the primary inoculum, henceforth continuing the cycle. Head blight is a fungal disease caused by Fusarium species. The symptoms for grains are observed on the head and neck of the grains and causes bleaching. Within barley, the disease does not spread anywhere beyond this. During humid conditions, orange spots can also form on the spikelets. Head blight disease grows most effectively in wet and warm weather during the maturation and growth periods of the cereal crops. Infection starts when spores of Fusarium species and on barley crops. The crops are most susceptible from the early flowering stage. The second most vulnerable stage of infection is the soft dough stage, where developing grain kernels are still soft. This results in the formation of tombstones and mycotoxin production will occur. These spores can also infect the roots through the soil, or with seeds that have been infected with head blight. This will damage the crop throughout its development cycle, therefore reducing productivity more so than infection of the grains alone can do. This is caused by a reduction of nutrient uptake. Early infection of the crop also increases its susceptibility to disease. Also, there is increased uptake of mycotoxins from the soil from head blight contaminated soil versus uncontaminated soil, which will increase the likelihood of the grains being rejected when being selected for further production. The scope and overarching aim of this project is split between 2 goals. The first goal is to create a mycotoxin predictor system at the early stages to understand when it's best to harvest a crop via a mixture of histology and PCR technique during a time when mycotoxin production is undetectable. PCR is a technique that can be used to detect RNA expression, and histological analysis can be used to take a portion of infected crop material and can be used to estimate the stage of infection, and combined can potentially use to estimate the current mycotoxin levels within the crop. The next goal is to design a novel mulching system to manipulate the environment of the soil to create an environment that inhibits mycotoxin production whilst maintaining the soil microbiome using a mulching system. In the microbiology side, I successfully managed to establish ergot on agar plates from using sclerotia and managed to grow samples in nutrient solution, and I created different potential solutions to infect barley crops. These samples have been used in microscopy experiments as well. The purpose of microscopy is to test my skills in microscopy ready for the histology trails of my post-infection crops, as well as work out if I were to place the infection solution in different conditions which will be most likely to infect. I have not yet attempted histology. However, I have achieved microscopy using the fungal samples I grew on the plates. I flooded the agar plates with sterilized water and mixed it and pipetted 5 microliters onto the center of the slide and then took images immediately shown on the top right. Next, I left different samples over a day and left them in room temperature, light top right and dark bottom left conditions, and another slide in the fridge bottom right to detect changes in morphology. A change from hyphae to a candida-like structure was detected in the fridge, where I plan this week to further see if any changes take place over several days this week. The fridge results do indicate that they are more likely to infect more effectively because, as seen in the ergot infection cycle, these can infect cereal crops in that form. Compared with the hyphae form the spores stuck to in other environmental conditions. The variety of crops I am using is Mordex. The crops I have grown within the environment chambers have successfully grown, however, infection did not take place with these generation of plants, therefore I have to wait until mid-May early June to try again when the crops hit the right point to try and infect. I have attempted to infect the crops with ergot spores using several methods, including brushing with a paintbrush covered in a bag to keep in moisture, paint brushing without a bag using both an oat nutrient solution spores along with spores made from mixing the plate with sterilized water. My next attempt on infection will involve a spray as recommended by Neil, another one of my supervisors, at an earlier date, as with some additional training I have found out I may have infected the crops too late. It had to be before the grain started forming, but just when the grain stalk starts growing is the ideal point of infection. Otherwise, a natural infection does not work. The goal of these infection trails is to test infection in different environmental conditions such as light levels, color, temperature, and humidity levels, as there has been research done regarding on how secondary metabolites, which mycotoxins are a part of, that influences their production. I have learned how to do fungal DNA extraction using a mixture of the Plant DNA Easy Kit along with the use of chloroform, phenol, and a specialized plant buffer at the beginning to break down the cells before having it washed through. I have also successfully identified several primers linked into the production of ergot alkaloids. The gene names are seen on the top of the diagram to your left, using a mixture of literature research to identify which genes and the fungal database to find the genes and primers if the primers have already been identified in other papers. PCR calculations have also been done regarding the different quantities I need to add to the PCR for synthesis, including the timing to make my own mixes. However, have not been yet successful when it comes to producing an reliable image to confirm the presence of the gene. Once I can perform it reliably, I will attempt to use it for RNA PCR to identify when the mycotoxin production genes switch on, to identify when the mycotoxin production takes place during infection, to see if the environment influence when and if this occurs during infection in different conditions. This can be used as a potentially reliable way to figure out when to harvest the crops early, as it would be better to harvest slightly less mature crops for use than more mature crops that are unusable for human consumption. The same experiments will be ran with Fusarium head blight, however we do not yet have permission to use it yet, as Fusarium species have an added risk of being in potential human pathogen in specific stages of its life cycle. Therefore, experiments are pending lab manager permission. Once an infection has been successful and sclerotia has been formed, mass spec will be used near the end of infection to measure the level of mycotoxin production that took place within the sclerotia. I also will be looking into post-infection using the sclerotia by putting them into plant pots to have them enter in sexual reproduction phase to do similar measurements in changes in mycotoxin levels using mass spec.